So extra amino acid is definitely there. So basically this is a diamino monocarboxylic acid. Okay. It is not monoamino monocarboxylic. It is diamino, and she correctly pointed it out that extra amino group is there. And that is the reason it is a basic amino acid. Okay, and because of the structural difference, one is arginine, one is lysine. So this slide was conspicuously missing, and from that time, my brain was not functioning well. What that slide have done basically. Second properties of amino acid, 3 points I missed, 3 slides were missed. I have shown you that these reactions due to carboxyl group and amino group. Can you hear? Last bench? You cannot. Now are you able to hear? Okay, with this loudness, then I will speak. So in that group, in the reactions due to carboxyl and amino group, I have missed decarboxylation, amide formation, and formation of Carbamino compounds. These are the 3 points which I could not discuss yesterday. So first is decarboxylation. See, this is the amino acid structure. Now if you have a decarboxylation, means carbon dioxide is removed, you can see it will get converted into this form, and this is called an amine form, because COH group is no longer there. So from these are the 4 points, you just note down this one. Histidine getting converted to histamine, tryptophan to tryptamine, tyrosine to tyramine, and glutamic acid to GABA. GABA is an important biomolecule. You have to read a little bit in detail so that if there is any short note, you can answer. From time to time, this particular glutamic acid or GABA will be told, and histamine will be told, not tryptamine and tyramine. So histamine will be required for all allergic manifestation for which there is an itching, and GABA is basically a neuroinhibitor. It inhibits the brain. That function is there. So you have to know that thing. So this is decarboxylation. Second is amide formation. Means extra amino group is being added. So I told you from glutamic acid, this is the structure of glutamic acid, to which we have added one amino group. So this COH group is no longer there. It is forming a bond. It is causing glutamine. Similarly, for aspartic acid, it will be formed that asparagine. Extra amide, that under the classification you see that amide group is there. So that comprises glutamine and asparagine. Okay. Then another important one is Carbaminohemoglobin. See, in the body, through the hemoglobin, oxygen is transported to the tissue and carbon dioxide is taken back to the lungs. The compound which is formed with hemoglobin, carbon dioxide, it forms a Carbamino, and you can see the amine groups are there. 4 molecules of carbon dioxide gets attached to form Carbaminohemoglobin or carbohemoglobin or carb hemoglobin. Basically what happens, in hemoglobin there are 4 globin chains, 2 alpha and 2 beta. Obviously this is a protein molecule, so it will have a good number of amino acids, and through that amino acid that carbon dioxide will get combined. This name is called Carbaminohemoglobin. In contrast, this is a reversible reaction, means in the lungs it will be able to release the carbon dioxide so that we can exhale. But carbon monoxide poisoning is a dangerous thing, and it happens in the factory, and there are usual deaths because that is a stable compound. The name of that molecule is carboxy. Although carbon monoxide name is there, the compound is called carboxyhemoglobin, but here it is carbon dioxide and name is Carbaminohemoglobin. Okay. So all these points of properties of amino acids are covered. Now in your syllabus, this first part I have covered so far. I think all of the things I have discussed in detail. Now in second part, then we will discuss classification of proteins, structural organization, functions and clinical aspects. So classification of proteins, under that classification function is there. On functional basis the proteins are classified. So functions will be covered in the classification part, and during function it is related to functional aspects. So clinical aspects will be invariably with their function. So I will discuss a little bit about the clinical aspects, because you see we are in the body, we are having almost nearly 80,000 to 1 lakh proteins. I mean, it is impossible to discuss all those things. I will tell you some of the important proteins. Rest of the proteins might be covered in plasma protein classes separately. And in small group discussion, of course, I will discuss in detail. This is basically a glimpse or overview of what are the clinical aspects. And these, what I am be telling, only a few slides, that will be clinically relevant, and some of the things are real picture which I have also treated. So protein, the term that in the book you will see that it is, the name has come from the word proteus, means primary. It is of paramount importance. Every organ, every body wants that it is important. So do you think that glucose is not an important thing? If the potassium level is low or potassium level is high, heart will stop in diastole. So everything is important. It is not that protein. All the things which are present, even the metals, even the vitamins, they are all required for optimal functions. So it was named in the earlier, it was a proteus. From that protein what has come. And only thing is that 3/4 of the body weight is composed of protein. And I told you categorically that 16% of the weight is there due to nitrogen. So why nitrogen? Amino group is there. Without amino acid, protein cannot be formed. And it is the nitrogen which is present in the amino group, and that is the reason nitrogen is very specific for amino acid. So using 20 amino acids, so minor components of course are there. Sulfur protein, I have told you, sulfur containing protein. Phosphoprotein you have seen in the milk. So using 20 amino acids, a variety of proteins to the extent of, there is estimates of 1 lakh to 4 lakh proteins. 1 lakh to 4 lakh proteins are produced, but there is an average in human being. By and large, 80,000 to 120,000 or 1 lakh protein you can remember the body produces. So we have been born and we die also. So proteins are also born, proteins also die. So what is the life cycle of protein? A life and death. Proteins are born typically at translation by using amino acids. And then it matures. As we grow up, we mature. It matures through post-translational modification. There are some modifications which are made And then some proteins require the similar group to be attached to act as a dimer or a tetramer or a pentamer, whatever way you say, or it may require some additional things like coenzyme or cofactor, metal ions to function optimally. So that is what is maturation. And then we all work and will take rest, so proteins will also at one point of time it will act, alternate with working and resting condition depending on the metabolic needs. Then of course at one point of time we will start aging from the time of birth. The protein will also age, and the aging occurs because of the oxidation and deamination. There are many anti-aging agents. See our skin, whatever way you see the skin of a baby and skin of a teenager and the skin of an old aged person will not be the same. Now anti-aging creams are used so that the skin looks younger. So the basic purpose is to inhibit this process of aging in the skin cells. So again at one point of time it has to die, and it will be dying by degrading into amino acid. The protein will be broken down. So it was formed with amino acid, it will die with amino acids only. And there is a good book written by Venki Ramakrishnan. He was awarded Nobel Prize in the year 2009 for extensive work on structure of the ribosomes, and this book was published in last year, and book is with me. So whenever you will feel free, after you read a little bit of biochemistry, because this is full of biochemistry, so you will understand that book, why we die, because we die definitely, but all the mechanisms of death are not known, and how we live, that is a very mysterious condition. Under what condition fertilized egg is there, but how do we live? How so many multicellular organs and such a good structure is made? They are very mysterious, but anyway. So lifespan of protein, just to give an idea that there is a regular turnover going on in our body. So old proteins are destroyed, new proteins are synthesized. As you can see in the life cycle, there is life and death. So once the protein is dead, that means new protein has to be synthesized, and this is mainly applicable for enzymes. So half-life is highly variable. I am giving you one example. This ornithine decarboxyl is one enzyme which half-life is only 11 minutes. In the animal kingdom also, mayfly, it can live for a few minutes to only 1 hour. Similarly, glass sponge is there in the ocean. Its lifespan is up to 15,000 years. That is the longest surviving animal, and that is the shortest one. And human beings, as you know, this lifespan is variable. So similarly, here it is there. So insulin only a few hours, but you know many diabetic patients are taking insulin, and it can not be given so frequently. That's why its longevity is increased by using one thing. One is protamine is a protein which increases delays its absorption, and zinc is added also to prolong its life, so that at least after 6 hours or 12 hours this might be required to be taken. So increasing the life expectancy of a particular protein, something is added. Hemoglobin, of course it depends on the type. If it is a natural hemoglobin, normal RBC, usually the lifespan is 120 days. But if there is an abnormal protein, obviously the lifespan of RBC, or due to any disease, if the lifespan of RBC is decreased, then the lifespan will also decrease. So one thing I am telling you, lens protein by and large remains unchanged throughout life until cataract formation. During cataract formation, as you know, in the old age, the lens becomes opaque and cataract surgery is required. Because the lens degrades, lens age, protein disintegrates and that forms clumping, and that is the cause why the so-called transparent lens becomes opaque. That's why the old age cataract removal is required. So, lens protein that is called a crystalline remains unchanged almost till it becomes, having a cataract changes. So, classification is based on basically 4 points. One is function, one is composition and solubility, shape and nutritional value. This is, I have checked with 2 books, they have written in the same way, so that I am showing this one. In our time, only one composition was there that was related to this composition only. That was only one small part, that what part was written during our time, that I will show it to you. And shape and nutritional value. Although peptides are not proteins, so for complete information, I will include some of the biologically important peptides. It can come as a short note, or bioactive peptides. See, biomedical importance of proteins. Why? This includes basically the functions. You note down there all the points. One major function is structural. Structural function. If we are living in studying in this hall, if the structure is not there, the actual function will not be formed. So, in life or in nature, forms follows function, remember. If it is not in a proper form, it will not properly function. So, structural function is support of building blocks. As I told you, amino acids are the building blocks for proteins. So, protein forms the building blocks, and collagen is the most abundant protein. It is present in the bone, it is present in cartilage, it is present in skin. The most abundant protein in the body is collagen. And you have to read, you will be taught 1 or 2 class, maybe I can take also on collagen itself. So, that is the vast subject. And you have to know in detail, because without collagen, healing will not occur at all. Similarly, elastin and laminin, they are all present in the extracellular matrix. Once something is injured, that means not only the cells, but the extracellular matrix are also damaged. So, that needs to be repaired. So, that part is there. Second, functional aspect means regulation of body processes. What are those? You try to note down if you can, otherwise I will show you at least one more slide, so that some points I have deleted deliberately. One is membrane proteins. Cell membrane has been told. So, what you have read there? Pumps were there, ion channels were there, transporters were there. So, they are proteins from all those things. So, that membrane proteins are there, they are acting as a carrier or they are acting as a receptor, and in course of time you will read each point very clearly. The sodium potassium ATPase pump is the most important pump, and that it uses a maximum energy. That uses a maximum energy, sodium potassium ATPase pump. So, transporters are there from outside the cell to inside, or from inside to outside. The transporter protein will be there. Channels, ion channels, chloride channels, as I told you, del F508 mutation is a chloride channel. And carriers will be there, receptors will be there, because when a hormone comes, on the surface of the cell, all the hormones cannot enter into the inside the cell, particularly steroid hormones. The protein hormones being bigger in size will act on the surface and it will send signal inside the cell that you have to work because I have come. Which one, Bob? In the bone and skin. Animal. We are talking all about human beings only. When there is any bacteria or plant, then that will be written specifically. Because if I go on speaking about all the living beings, then that will be difficult. We are a medical student, so most of the things will be related to human beings, but we are living in nature. We are exposed to bacteria. We take plant foods. We take animal foods. That can cause some problem or diseases. Again, so in those cases, I will specifically tell about these things. Okay? Now then, catalytic function. All enzymes perform catalytic function. That is one function aspect. Practically all enzymes are protein in nature except tRNA. There is RNA. Don't tell that RNA is an enzyme. That is the one exception. Okay? So that exception will be told what role it plays. Everywhere there is some exceptions are there. So regulatory role, hormones play a regulatory role, definitely. Yes? Exception RNA is an enzyme. RNA as an enzyme. RNA is basically nucleic acid, but it can act as an enzyme and that is an exception. But don't tell that exception at the very outset. Give an example of one enzyme. It's not RNA. You have to name one enzyme. You just write salivary amylase, amylase, lipase, whatever, chymotrypsin, trypsin, whatever you say, these are common enzymes. Okay? Glucokinase, hexokinase. I mean, practically all, that's why I have written. Practically all. That one exception is there. Maybe some new part will be discovered as an enzyme. So practically all enzymes are protein in nature and they perform catalytic function. Then regulatory function, hormones, even the enzymes perform regulatory function. Enzymes are regulated. As you have seen that enzymes alternate resting phase and activity phase. So these are regulation basically. At what point of time it will not function, at what point of time it will function. Okay. This is just an exhaustive, it is not indicative list, not exhaustive. Then defense immunity, immunoglobulin. So if you see the immunology is a branch which is taught in a DM course for 3 years post MD. So that cannot be covered. Immunity deals with one subject, full subject, that is called immunology. And immunology DM course is there. And DM course is of 3 years after MD. So that cannot be covered in one class. That's why I told this is indicative only. So one class I will take on immunity, 3 classes I have taken on immunity itself for you, last year. So transport, hemoglobin you know, it transport protein. Similarly, albumin also transports, it's a donkey molecule you can say. It carries many things. Binding proteins, there are several binding, thyroxine binding protein. Thyroxine is produced here, so that needs to be transported to the tissues. It is not that soluble, it is transported through thyroxine binding globulin. The contractile protein, movement, body, actin and myosin. These are contractile protein. Sensation, on light we see something. that is rhodopsin. Rhodopsin is again opsin plus 11 cis retinal. So that is also an protein. Huh? Contractile. Rhodopsin visual sensation. Rod cells, yes. In the rod cells, rhodopsin is there. In the rod cells, rhodopsin is there. This is opsin plus 11 cis retinal. That was the composition. I am telling you just to note down at this stage. This is basically an indicative, means a preliminary information. You will be told in detail about this rhodopsin functions. There is a world cycle. Remember, since you have raised this question, write down in the bracket. World cycle. World cycle is there. WALD. World cycle is there, and that is that visual cycle is there. How we see basically. Fluid balance. Most important thing is albumin. Albumin maintains body's fluid volume. It is an osmotically active substance. Genetic. DNA packaging, nucleosome. Around that histone proteins, the DNA is packed. So that packaging is also important. So the histone protein is defective. So again, that nucleosomes will not be formed. Providing energy. Normally, proteins we do not take for energy. We take energy from carbohydrate and fat. Can anybody tell how much energy is produced from protein? How much energy is produced from protein? 1 gram of protein? Huh? 4.5. 4.5 kg? Calorie unit, always you say unit. Without unit, there is no value. Okay? So normally we do not take because carbohydrate is a cheaper source of getting energy, and unlike protein, unlike carbohydrate or fat, there is no storage form of protein. That is one important aspect you must remember. In carbohydrate, if you take, it can be stored as a glycogen, excess amount. If you take fat or carbohydrate, it can be stored as adipose tissue fat, but there is no storage form of proteins. If you take excess amount of protein, which happens during party, the extra amount of protein will be simply broken down into urea and it will be excreted through urine. So, until and unless energy depletion is there, energy from proteins are not usually taken. We take adequate amount of carbohydrates and fats from where our energy requirement is met. So, don't think you take too much of protein means too much of body weight. No, it will not happen. No, it will not happen. So, there are several misbeliefs. That's why I have shown you on the very first date that Sharir Ghire Samskar. For energy, I repeat, taking energy, you take carbohydrate. But fat also you have to take a little bit of restriction. Carbohydrate you have to take restriction. By and large, I am telling you, the food composition, the entire energy should come 60 to 70% from carbohydrate, 25 to 30% from fat, and 10 to 15% from body protein. That will be the ideal composition of a diet. But if you take that, I will not take rice, but I will take meat only or chicken. You can decrease the rice. That protein which you are taking can be used utilized as an energy. That is altogether a different issue of not today's discussion. But that is a costly item. See, taking rice will be much cheaper. That cost of the rice or cost of roti chapati will be much cheaper than the cost of a protein. Storage. I am telling you, there is no storage form of protein in the human beings. Plant can store. But there is one thing called ferritin. it stores iron, not protein as a source. Suppose we have not taken any food. Now we are having glycogen as a storage form of carbohydrate. Now glycogen will be broken down. Once glycogen stored is depleted, then the fat stored, that triglyceride will be broken down. Once that triglyceride part is also exhausted, then the skeletal muscle protein will be broken down. Okay, that stage is different. So we don't have a storage form. So of that protein, that you must remember. So abnormality of the protein structure, of course, will cause disease. Abnormality in the formation as a structure, or if it is produced in the less quantity, or if it is produced in the more quantity, both are dangerous. So one point I would like to say, that anything which is detrimental to health is a poison. Water is a poison to a person who is having edema. Water is a poison in a chronic renal failure patient, kidney failure. If you give excess water, it will cause excess volume formation. So sugar, the glucose, is a poison for a diabetic patient. If he takes excess of glucose, of course that will have a deleterious effect on the body, that is a poison. So anything that has a deleterious effect on the body is a poison. It may not be a poison to you, but it will be a poison to diabetic patients. So functions, apart from those functions, those if you cannot remember at least these 6 points. One is structural part, that is collagen and elastin. One is contractile, that is actin myosin. Transport, hemoglobin. Myoglobin is not a transporter, wrongly I have written. Albumin, because myoglobin is fixed within the muscle. It doesn't transport. Albumin, transferrin, hormonal. There are many protein hormones are there. The list of protein hormones I have written only 3. Genetic histone proteins are very much important, and protective immunoglobulins will be there, interferons are there. And then I told you about immunology. I have taken 3 classes, so this year also I will try to cover all those things. So this is a chart which we have read during our MBBS class as classification of proteins. That simple protein, conjugated protein and derived protein. So at least you must tell this thing about the composition and solubility. So albumin, simple protein, forget about all these things. You must tell at least albumin and globulin. This is an example of simple protein. So simple protein will be albumin, globulin. Protamine is an important protein. I will say to you. Prolamine, lectin you may not remember. Scleroprotein is important. So albumin, globulin, scleroprotein you remember. Protamine is also present because it is present in the sperm. It performs a function which is of course required for reproduction. Conjugated proteins means protein is conjugated with something else. Protein is conjugated with something else. What is something? If it is conjugated with glucose, it is called glycoprotein. If it is conjugated with lipid, that is a lipoprotein. Nucleoprotein, and then chromoprotein means there is some colored substance like hemoglobin, like myoglobin. Phosphoprotein, I told you that phosphoric acid can be combined with a protein, so that will cause phosphoprotein, and it is present in casein in milk. So those who are suffering from basically osteoporosis in the old age patient, we usually recommend they take adequate amount of milk, because phosphate will be supplied and protein will also be supplied. So at least that ladies particularly suffer from osteoporosis after menopause. So if they want, I would advise calcium tablet, but many people are fond of taking natural foods. Then you take milk, but in a very high amount, and then that will be a costly one. Anyway, metalloproteins. some proteins require metals to function properly, like enzymes as a cofactor. Derived protein is something we are deriving from the proteins. So, if the protein is broken down, the first protease will be formed and then peptide will be formed. In books you can see the polypeptide name is there, but amino acid name is there, but definitely they are derived from the proteins. If you break down serially, protease, peptone, then polypeptide, then peptide, and at one point dipeptide, tripeptide, dipeptide, 2 amino acids. That will be the sequence of events. Simple protein. His name I told you albumin. At least you just tell the name of the albumin. What is the simple? You know the details of this thing. You note down all these things. Note down this part. So it is soluble in water. I am telling you, if you take pure albumin and pure water, and if you try to dissolve it, it will not dissolve. It will not dissolve. For albumin to be soluble, because albumin structure is very large, 66,000, 67,000. So it will not go into the solution, it will form a colloidal solution. So some amount of salt is to be added. So that is called salting in. If you add a little bit of salt, then it will get dissolved very easily. Albumin powder is not with me. If you purchase any albumin powder anytime, egg albumin powder, you bring it to the laboratory or you experiment with yourself. With water it will not get dissolved. Add a little bit salt, it will get dissolved very quickly. So albumin, although it is soluble in water, not in pure form, not in distilled water. So mature human serum albumin, molecular weight is 67,000, you can remember. Bovine serum albumin, bovine means cow. Bovine serum albumin is extensively used in the laboratory. You raised a question about this human being at the last bench. Your friend, you raised a question. Hello, not this one. You raised a question about human or non-human. Whenever there will be non-human origin, I will definitely tell. This is one example of bovine serum albumin. Bovine serum albumin is used in the laboratory. Okay, for experimental purposes, not in the human being, because it is antigenic. This cannot be given to a human being, but human serum albumin can be given. In the milk, we are having a lact albumin. This can be present in a human being, this can be present in other animals also. Egg is not present in the human being, so egg we consume. So in the egg there is a protein called egg albumin. And remember, another point you note down, there is the most abundant protein in the blood plasma is albumin. And it maintains oncotic pressure. Oncotic and osmotic, there is a difference. Osmotic pressure can be made by anything. All the ions, non-ions, whatever you say, glucose can also exert an osmotic pressure. Ions can also exert. But colloidal molecules, colloidal which are not in true solution, when they exert that atmospheric pressure, that is called an oncotic pressure. So albumin is a colloid substance. So albumin will exert a pressure which is definitely an osmotic pressure. But that osmotic pressure is called oncotic pressure. You can tell colloidal osmotic pressure of albumin, or you can say oncotic pressure of albumin. Both are synonymous. So it binds with the transport of bioactive molecules, many molecules. You will see during your metabolism classes that it transports fatty acids, it transports bilirubin, it transports hormones, drugs, metal ions. So I say albumin is a donkey molecule. You carry anything, you put anything, it will carry. It's a donkey molecule. Its approximate half-life is about 19 to 21 days, and that is used for at a drug delivery. So if you combine with a drug, so drug delivery, drug life will be increased because albumin is there, so it will slowly release the drug. And in clinically, if the albumin level in the body goes down due to any cause, either because of the synthesis, less synthesis, or over-excretion due to disease, albumin level is decreased, in that condition you can give albumin, human albumin. Or if there is extensive burns, you have not seen the burns cases, if the body is extensively burned, what happens? Blisters are formed. You have seen definitely the burns, small blister, but in case of extensive burns, huge amount of extracellular fluid will come out, and that comprises a good amount of albumin. So there will be loss of albumin. So immediate loss of very high amount of albumin, and that will decrease the osmotic pressure of the plasma, and that will cause problem. We do have many plasma expanders. Plasma expanders means there are many drugs which can expand the plasma volume. But when that is not available, or when there is real need, then only human albumin is given to burn patient, not otherwise. Similarly, if there is a hemorrhage, huge amount of hemorrhage, blood loss is there, means along with the blood, plasma is also out. Now in the blood, 55% plasma is there, 45% is the packed cell. Now in the plasma, this albumin is there. So blood loss means plasma, albumin loss also. So normally when there is a blood loss, blood is transfused, not albumin. And if blood is not readily available, then the plasma expanders are given to keep the volume all right. Now when that is also not available, as a third option, second line option or third line option to make the patient survive, you can give albumin, but this is not a normal indication. Second, what is the major function? How did the body, this is one of the major function I told you, that it maintains the extracellular fluid volume. How it does? Basically, through the artery, blood is flowing. At one artery will be broken down just like a pipeline, water coming from, suppose Bolpur to this place. But the main pipeline will be there, and the room you will get the smallest pipe. So that is basically the arteriole. This arteriole is there. Now arteriole will break into capillaries. And then from the capillaries there will be venules will start. Blood will pass basically in this direction through the capillaries to this direction. Okay. Now as the blood flows from the big artery to the small artery, the blood pressure drops. As you know, normal blood pressure, systolic blood pressure is about 120 degree and 120 millimeter, and diastolic is 70 to 80. Now that pressure will not be there at this level. Whatever the pressure is there, the Bolpur from where the water is coming, the pressure in the pipe will not be the same as what the pressure is there in your room. So this is your room, remember. And this is the other structures of the room, basins, toilets, the pressure will not be the same from the main pipe. So basically, but the water is same. So the albumin which is present in the plasma will not come out. Here what will happen, because of the fenestrated structure, blood pressure will be there, that will cause a hydrostatic pressure, and that will try to drive the fluid out from the vessel into the extracellular space. This is extracellular space. Extracellular space or extracellular fluid you can see. So this plasma, while passing through the capillaries, because of the hydrostatic pressure, it will force the plasma, that water component, to pass through this structure. Now it will be prevented by albumin. because albumin will exert colloid osmotic pressure, it will try to keep the water in position, not only albumin, other proteins also. But albumin is the most abundant protein. Albumin will try to keep that solution in that thing. So here the blood pressure is about 35 millimeter. Of mercury. And colloidal osmotic pressure is about 25. Okay. So there is a difference of only 10. And because of the 10 difference, some fluid water will come to this place. Okay. But this albumin, which cannot come out because of the larger size molecule, it will pass through this structure and it will again maintain a pressure of 25. But now the blood has passed through capillaries, means the pressure will again drop, and it will drop to almost nearly 15 or 17, you can say, is BP. Now here BP is lower, colloidal osmotic pressure is higher. So whatever extra amount of fluid has been delivered in the extracellular space will be again sucked back into the venular end. So that's why we are not having any edema or extra amount of extracellular fluid. Do you understand? Now if the albumin level is low, স্যার চলে এসছে এই স্যার চলে এসছে আচ্ছা I'm stopping it here. So if there is a small amount, I mean the albumin level is low, that means the colloidal osmotic pressure will also be low. Suppose it is from 25 to 15 to 10 to 5. Okay, just for example, because of the disease process. Blood pressure will remain same. So this gap, what you are seeing here, the difference of 10, this 25 will again decrease. So fluid will not be sucked back into the venular end. So that will cause fluid accumulation. Okay, that will cause the formation of edema, and because sir has come and this. Otherwise, I'll show you. This part I'll discuss tomorrow again. So this is the structure, I'll show it to you. This will happen. See, now you see the pitting edema. You have seen definitely swollen faces. Normally we are not having. Why we are not having? Because we are having adequate amount of albumin. Now that if albumin is decreased progressively, now when that patient might come, when the early stage, you will not get that much dipping. But there is slight amount of dipping is there, means the extracellular fluid is there. So very early stage, you have to check all the body whether there is an edema or not. So then you can detect the disease early. Okay. So with this we conclude. Tomorrow I will discuss, maybe I will be required. Is this speed all right, or should I go a little faster? Because then, okay, in SCT class, I'll go still slower, so that you start interacting.